Speaker is Mark McCarthy. He's the Executive Director of Human Genetics and Principal Fellow at Genentech, where he leads efforts to use human genetic data to inform disease biology and therapeutic discovery. He was previously served as the Turner Professor of Diabetic Medicine at the University of Oxford, and he's led many international consortia in identifying hundreds of loci involved in type 2 diabetes and related traits. And that's where Mark has been collaborating with Mike on many of these large-scale GWAS. So, welcome, Mark.
Terrific, great. Yeah. Thanks very much for the invite to come here. I was so pleased to see you, not see that he was retiring, but seeing that there was this symposium to recognize his 70th birthday and his quasi-retirement, as I perhaps should phrase it. And delighted that I was able to make it. Sorry I couldn't make it yesterday. I was in London on Wednesday, but I was able to get here yesterday evening. So yes, my history with Mike goes back quite a long way. I'll tell you something about that. And I'll try and give you some sense of the way that the things I've learned from Mike, the work that we did together has kind of powered me through into my new role at working in industry. So for those that don't know me, I'll just give a little potted history. This isn't about me, it's about Mike, but I trained as an endocrinologist, was dragged kicking and screaming into the lab to do some research because I really hadn't done any research, and it was considered a really reasonable thing if you wanted to get a good job in the UK as a medic to do some research. And what was going on in the lab was type 2 diabetes research. So I went in the lab, started pipetting, doing gels, a pretty traumatic experience. It was traumatic for me. It was probably a darn sight more traumatic for everybody else in the lab. And I was ushered out of the lab and onto the computer, and it turned out that I could actually do some of this computational stuff, segregation analysis and so on, reasonably well. I was put in touch with Newton Morton, who many of you will know, and I started going down a computational track. I was lucky to spend a year with Eric Lander at the Whitehead then in the mid-90s. I returned to a job at St Mary's in London, part clinical, part research, and then did less and less clinical as the years went by, more and more research. I moved to Oxford, and then 7 years ago, gave up the sunny climate of for the windswept plains of Northern California. And the colors haven't come out quite right, but this is the sort of Boehnke mapping on top of this, right? So the time in Oxford were really peak Boehnke for me, and worked very closely with Mike over decades there. I'll say something about it, but it's lingered on, and that's what that yellow is meant to show. I was one of the nice things about giving this talk is it gave me a chance to go back and ponder my previous existence, but also Mike's previous existence. And I thought about, well, what's probably the first time that I ever came across Mike? And it's probably in those years when I was thrown into a collaboration with Newton Morton. Newton Morton is a lovely guy for those that knew him, but as somebody said, you really need to use a friendly interface, because he was talking in language that was definitely not a language I fully understood. So, but I started poring over the literature, and I'm sure at that time I would have come across some of Mike's early papers that started to educate me in statistical genetics. So I'll say a few words about what I do at Genentech, but then I'll go back and sort of ponder the previous few decades. But I think all of you know that drug discovery is a pretty difficult place. Many perfectly reasonable candidates that look strong on the basis of biology, plus or minus genetics. Very few of those make all the way through. Maybe lucky if it's 5%. Even when they get into the clinical phase, probably only 1 in 10 succeeds at becoming an approved medicine. And it's an expensive business with a very low success rate, and often can take you 10 or 15 years before you find out whether you've made a good hunch or not. And as many will know, that's not a great economic model, and people are looking for ways to improve that. And every year or two there'll be some new flavor that's supposed to improve the odds. It's AI now, obviously, that's very much being targeted. I'll say a bit of words about that. But as many of you know, human genetics is also emerged as really a pretty powerful technique and tool to bring to bear on drug discovery for all the reasons that everybody knows. It's in vivo functional biology in the species we most care about. The natural experiments of genetic perturbation that we explore have analogies to the clinical experiments of therapeutic perturbation we're trying to achieve. We have the advantage of unidirectional causality that can help with causal inference. And then most latterly, a whole series of studies have demonstrated that if you do choose targets that have got genetic evidence, that they're around 2 to 3-fold more likely to be successfully prosecuted through the drug discovery program. So it's in humans, it's causal, and it's been shown to work. And so my last few years at Genentech has really been involved in repeating this, showing this slide as often as possible to as many people as possible. And when my not very genetically favoring boss came back to me, it's Mark, I've just heard that targets with 2 to 3-fold with genetics have 2 to 3-fold the chance of prosecution, something I had been telling him every moment, every waking moment for the previous. He said, it's amazing. So I realized then my job had been done. But it does show that you do really have to repeat these things ad nauseum to break through. And the reason, of course, why human genetics is useful because it can contribute not just to finding targets and prioritizing targets. It can help you think about indication space and repurposing, give you clues to safety, help you identify biomarkers through interfacing with proteomics, for example. It can help you get to mechanistic insights, although, as we all know, that's tricky, and that's one of the areas where genetics perhaps has been disappointing over the last 20 years in that we find many signals but not always able to understand exactly how they influence the disease. And increasingly, the promise of precision or personalized medicine, being able to stratify disease risk, think about therapeutic response, and maybe even use genetics as a tool for doing more efficient clinical trials. So in terms of some of the themes that we're adopting at Genentech, there's a big emphasis. We're in California, after all. We're in the Bay Area. We have Aviv Regev as our EVP, who came from the Broad. So not surprisingly, there's a lot of emphasis on big data, and we eat AI for breakfast, lunch, and dinner. And so some of the big efforts that we have around doing large perturbation screens, doing large tissue spatial analyses, doing large analyses in cancer data sets, those three really to set up to build foundational models, to understand the embeddings, to get representation of disease, to be able to generalize outside the systems in which you've done these experiments, because you can't possibly, of course, perturb every possible cell line in every possible state with every possible gene, every possible compound. That's the data set you'd like to have, so you have to generate some parts of that and hope you can learn some rules that allow you to generalize beyond that. And then lastly, I've been bringing human genetics to the table to really start to unite all of these and think about ways in which we can build across all of these to understand the pathobiology of disease. So I thought what I would say in the rest of the talk is just go through some of the things that I've learned from prolonged Boehnke exposure and the lessons I've learned and ways in which some of that is carrying forward into the work that we're doing at Genentech. So first of all, most important thing, as you say, you choose your parents wisely. That's a pretty good thing in life. But in science, choose your collaborators wisely. And I've collaborated with many, many people and many in this room. But the triumvirate that we set up and worked on in the diabetes space for a decade or so was the most rewarding, I think, between myself and Mike and David Altshuler, who many of you will know. I think David was here last week. Did he show the slide? Yeah, I thought he might have, because I actually got the slide off him. I do want to tell you, this is a fake slide. It just shows you how good AI can be, because it's got the Michigan swag in there. It's even reproduced ASHG badges from 2019 and got our names on it. But I was able to dig out the original photograph that was manipulated to generate this II. And here. But actually, I didn't ask it to put the genetic drifters on there. And it came up with genetic drifters as the name of this super group that was reforming. So that's one shot as well. That was a very simple prompt. It's worrying. It's worrying. So that collaboration really built on what we heard about yesterday from Nancy and Mike's work putting the linkage analysis consortium together. And I really echoed everything that Nancy said last night about how it didn't need to have been that way, and if it hadn't been for your efforts and Mike's efforts to get a bunch of somewhat fractious people. In retrospect, of course, it does seem a little bit odd. It's a bit like the argument about bold men fighting over a comb. There we were all fighting to be the one to find a big linkage signal for type 2 diabetes, the major gene for type 2 diabetes. Don't want to disappoint anybody in the audience, but there isn't one. And, but it presaged the move onwards to other technologies. And I was fortunate to be working with folks in Oxford on doing GWAS for type 2 diabetes as part of the Wellcome Trust Case Control Consortium, and obviously Mike and Francis and many people in this room were engaged in doing the same in Fusion, and David and the folks at Broad working with Leif Group, doing something analogous again in another Finnish cohorts. And I think Mike and I agree it was probably about 2005 at the ASHG that we said, well, we hadn't got any results yet, right? It was still, we're, I suppose, a bit optimistic. We'd seen how AMD showed you could actually get hits in 100 cases and 100 controls, but was that going to be the exception rather than the rule? Well, it was the exception, but and we all invested pretty heavily, and we're feeling a bit exposed. And what if we did these studies and nothing came out of them? Well, wouldn't be great to have easy communication, ways to share findings even before we published so that we knew what was real, and maybe if a signal wasn't quite significant, we could corroborate it in other data sets. And that worked out incredibly well and led to those 3 papers back-to-back in Science, and maybe even somewhat unusually, we each of us, this is the table from the Fusion paper, we each of us quoted the results from the others to sort of build up those patterns of corroboration. So second, be flexible. Don't hesitate to switch things up. I'm not advocating the health and safety depicted in this cartoon, by the way. It looks a bit troublesome. That train's coming off the rails. I'm not quite sure why it's sitting between the tracks to change the tracks. So AI has a bit to learn. This is this linkage analysis consortium paper that work that was done. The paper only came out in 2008, I think, but the work was done probably in the early 2000s with Nancy and Mike leading it. Those log scores look pretty impressive, but I think we discovered that there was a lot of technical issues, the gene typing error and so on that Richard Watanabe mentioned yesterday. But I did want to just start by just saying, my God, I'd forgotten so many people involved and so much effort to include people, patients from so many diverse origins and so on, which is a theme I'll return to in a minute. Numbers were even pretty impressive for the day as well. This was all sort of SIB pairs and so on with linkage. So kudos to both of you for pulling that together. And I won't mention any names, but some of these people weren't the easiest people to deal with. So having thought that we'd maybe found some rare variant signals with the linkage, probably misleadingly, the decades from 2007 on were this really starting to enumerate all the common variant signals that were clearly emerging. It amazes me looking back in retrospect how quickly we were able to move to pull these meta-analyses together and get people to replicate it, and then started publishing these papers jointly. This is the one from 2008, finding another 6 signals for type 2 diabetes. So it was pretty confident that there were common variants. Those of you who remember the time will also remember that there was a lot of sniping from the sidelines saying, well, you're finding all these things, but aren't they terribly small effects? And they aren't really explaining most of the heritability, are they? And this whole concept of the missing heritability came up, and where was that residing? And there were certain protagonists that said, this is all in rare variants. That's what you're not seeing. So it was natural for us jointly to try and plug that gap, which we did, culminating in this paper in 2016, which again was a joint effort between our 3 groups, but many others besides that contributed, where we brought some exome sequencing and some whole genome sequence to the table, just to start to be able to explore those low frequency and rare variant space. Although power was obviously going to be pretty small, we were able to do some modeling there, comparing the empirical distribution of hits in low frequency and rare variant space that we'd seen in the analysis with different models, the second model being a rare variant, a predominant model where you would have expected to see hundreds of hits if that was the model explaining the heritability, and demonstrating actually the closest congruence to the empirical data came from a model which limited the rare variant contributions to about 25% of heritability, which is actually not too far off where it's ended up in most estimations, probably about 10–15%. So another thing that's pretty clear is that sample size matters. And I plotted this out. I hadn't bothered to plot this out before, but I plotted it out the other day, which shows the number of significant signals in the various studies we'd done over the years together, plotted against the number of cases on a log-log scale, as you can see it, and it's very nice and linear, and would have been good to have this for grant proposals. So, culminating in the most recent paper from the group. Actually, Mike and Dave and I have sort of stepped back from this now. There's a whole new cadre of fantastic investigators who are carrying on this work, which is one of the things that's immensely gratifying, to feel that you can leave the stage and the world goes on, and there are smart people doing even better things with the data in subsequent phases. But 400,000 cases to 2 million controls and over 1,000 hits for type 2 diabetes. And clearly we've got a lot of opportunities to expand beyond this. This is just a map of some of the cohorts that we interact with or have interacted with or are thinking to interact with at Genentech. So not a complete list, but there are clearly growing numbers of databases from biobanks and healthcare systems across the globe now. But return to a theme that I picked up earlier. I mean, diversity is the fuel of genetic discovery, and that's something we've not been particularly good, despite best efforts at engendering. So much of the data, as you know, still comes from European descent individuals, and that's true really for the internal data we have access to from our trials, which are still, despite best efforts, often strongly Eurocentric in terms of the representation in our trials. But again, I think credit, Mike was, as I showed with the Linkage Analysis Consortium, was always thinking in that direction, and he'd been exploring collaborations in East Asia, and I'd been exploring collaborations in South Asia, and so on. So we were able, as the group coming together, to start to build the first multi-ancestry analyses for type 2 diabetes, a reflection of the fact, of course, that diabetes is common, and it's common across the globe, and therefore there were, within even quite small biobanks, appreciable numbers of individuals with diabetes that could be used for these analyses. So this was a paper from mid-2010s, I can't remember exactly, where we're starting to at least build up some representation from non-European data sets. And that expanded in the most recent paper that I mentioned here, but still, it's still 60% European effective sample size, 20% East Asian, and really the rest of the globe, only in the remaining 20%. And big gaps in Africa, big gaps in South America, big gaps actually in Australia as well.
Can I stop for a second? Somebody has their phone on and we can hear it. Could everybody check
It's me talking, sir, Laura. Now, anyway, I mean, things are moving in the right direction, right? Again, if you color in the major ancestral group in the various data sets that I know about that we're interacting with, there's certainly much greater representation possible from these, with a big caveat, black box warning. I am a farmer, we know about black boxes, right? Reducing the complex spectra of individual genetic diversity to simple ancestral and/or continental categories is seriously problematic from scientific, historical, ethical, and operational perspectives. We all know that, but we still tend to use these labels for the absence of a better lexicon. I hope we can do better in the future. I mean, in Genentech, we've particularly focused in Africa. Many of you'll know that, all of you, I'm sure, will be aware that most of human diversity is on the African continent. A sixth of the world's population, a quarter of the world's population in generation, 25% of global disease burden, a shift from infectious disease, non-communicable disease, but trivial proportion of individuals in both clinical trials and in genomic data sets come from the African continent. In fact, that percentage is going down because the growth of data sets from elsewhere has outpaced even the meager contribution from African data sets. So that's something we've been able to invest in from Roche and working with other pharma partners to try and see if we can contribute to efforts to rectify that. So we've been established a consortium which is in the process of generating 50,000 diverse African genomes, most of that sequenced on the African continent and analyzed on the African continent, because we think that's the way to do things. The days of helicopter genomics are long gone, because it's important to build capacity and build opportunities for genomics to be done on the continent. From that, we can go on to building out some biobanks. We're already collecting about 100,000 with other pharma partners in South Africa, and then start layering on multi-omic data. So we can get some of the way to fill that really quite embarrassing gap and seriously detrimental gap in availability of data from the African continent. Data transfer conjecture. I think this came up yesterday as well. If I had £1 or $1, now I live in the States, for every time I'd heard one of these arguments, right? Diabetes is too heterogeneous. The way to find signals is to deeply subphenotype and then do your genetics on those more homogeneous data sets. That was very common. And the answer to that is, well, yeah, go for it. Let's see how you do. I mean, our attitude was very much let's pile them high, lump, and then split later, right? Rather than split first. I alluded to the second one just earlier, common variants aren't explaining much of the heritability. It's all really down to large variants of large effect. Well, no, it's not. The best way to find signals is to match cases and control as far as possible for extraneous factors. That seems kind of reasonable. I'll show you a couple of examples where that didn't pan out quite as you might have expected, and that these GWAS association signals of such small effect that they have no value for therapeutic identification. And again, that's not borne out by some of the data I showed you before. But I think Mike's attitude and certainly one that was reflected in the rest of us was, well, let's go out and gather data wherever possible to test these assumptions. We can cogitate, we can hypothesize, we can have arguments in bars, we can have arguments in coffee shops, till the cows come home. At the end of the day, it's data that's going to tell you how you should go forward. So on the question of matching cases and controls, one nice story comes from those first 3 GWASs that we were doing back in 2006, 2007. And it was really reassuring when we started trading results that most of the signals that we got in the Finns and in the Brits across these 3 studies were showing up. And that was, we didn't know it. We're so worried about all the potential technical errors and artifacts that could be there. So it was just great to see that. But there's this one that was misbehaving, which was the FTO signal, which we saw in the UK data sets, but really had very limited signal in the other 2. And we worried about this. And we'd got the results, and we were sharing the results sometime in December of 2006, as I recall. My sister lives in Toronto. So we went out for Christmas. We flew out on Christmas Eve to Toronto to spend Christmas with my sister. That's just me, not me and Mike and Dave. That would be a bit weird. And as you do when you, I woke up like 2:30 in the morning and I couldn't go back to sleep. And I was, sad, but this came into my head, and I thought, what if I know that they controlled their case and control for BMI, right? Because they didn't really want to have all those BMI effects get in the way, and we hadn't done that. So what if maybe this is obesity? And it was so at like 4 o'clock in the morning on Christmas Day, Toronto time. I get on the laptop and send out to all the teams, which is a bit depressing thing to do on Christmas Day. But I said, most of you are not going to see this email. But if anybody wants to just run analysis to see whether this is associated with BMI, and then there was one Sado who shall be nameless. Within 30 minutes, got back to me and said I've logged on, and it's associated with BMI. So yes, that's how we found what was the first, I suppose, confirmed common variant signal for obesity, but because we hadn't matched for obesity. And a second example of that, which is more recent, this is some work from some colleagues at Genentech. This relates to diabetic retinopathy, which is one of those traits that really hasn't read the textbooks, right? It's very heritable. There have been very large studies done, and yet there are very few, if any, reproducible signals for diabetic retinopathy, which is a bit weird because many other equivalent traits have got hundreds of loci by now. So many possible reasons for that. One of them is maybe there's some heterogeneity. And we're fortunate to be sitting on this data set with a subset of diabetic retinopathy called diabetic macular edema, which is about 20% of DR. And because we'd been developing drugs for wet AMD, and the pathology of DME and wet AMD is quite similar, and we had an agent, faricimab, that was approved for wet AMD. We started running some trials, which subsequently got it approved for DME as well. And those trials meant that we had like 1,500 cases of DME that had been recruited in these trials with very strict criteria, and we'd done WGS on them. So we had 1,500 cases with WGS, mostly European, 10% African. And we compare them against, we don't go sequencing normal controls, because that doesn't make much sense, but we used other WGS trial participants with orthogonal diseases as the controls. So we thought, well, we'll just run a GWAS for this. Let's see what's the worst that can happen. And we were pretty surprised to get this huge signal over ApoL1. Now, many of you know about ApoL1, but here's a signal, a coding variant ApoL1, highly significant. Most of the signals coming from the Europeans, but it's even present nominally in the African descent individuals. We were able to replicate it in 2 other data sets. So there was no doubt that there was an association of this coding variant ApoL1 with diabetic maculopathy. So what's going on? I mean, most of you will know ApoL1 because of the protection against trypanosomiasis that has caused it to rise to quite high frequency in African individuals and led to protection against a variety of kidney disease. So a number of possible explanations for what might be going on. First of all, were we just lousy in choosing our controls? No, we could map that out. The second thing that the reviewers weren't very happy with was everybody knew that you had to do your studies in diabetics with and without retinopathy, right? The cases should be the diabetics with retinopathy, and the controls should be the people with diabetes without retinopathy. That makes some sense, but we hadn't done that because our controls came from people with a variety of other diseases. But of course, that's pretty easy to exclude because we've got these monstrously large type 1 diabetes and type 2 diabetes GWAS, and you could say, was there any signal in ApoL1? No, flat as a bat. And the other one could be, maybe this is something to do with that renal story and the risk of renal disease. We know retinopathy and nephropathy go together. Well, we could test for that as well, again, in very large GWAS. And there was no association of this variant with DN. It was associated in African Americans, but not in Europeans. So what's going on? And to understand what's going on, you just have to look at the haplotypic structure of APOL1. These are the two haplotypes, the G1 and G2, which are African-specific, and it's these variants up at the 3' end of the gene that are responsible for the phenotype that gives you the protection against trypanosomiasis and the renal disease. But those are absent in Europeans, so they can't be explaining those associations in Europeans, which are coming from this variant here. You can see how this variant is picking up the DN in African Americans, but the only real explanation of this is that the susceptibility for diabetic maculopathy is actually coming from a different site within the gene. And that's led to interest. Obviously, a number of groups, including Vertex, David's company, have been looking at APOL1 therapies for kidney disease, and there's some expectation that you might be able to repurpose that for diabetic maculopathy. How am I doing? Oh, I've got another 2 or 3 minutes. Genetics only gets you so far. I think one of the things we learned pretty early on is that you need to bring in other data sets to really go beyond just putting pins in the map, in the genomic map. If we're really going to get to function, we need to bring multiple types of data together. And working with Mike, that was really one of the themes of the work once we worked out how to do these meta-analyses. One of the data sets that I'm most proud of came from this paper led by Ben Voigt, jointly on behalf of the 3 groups in 2010. And what he did here we had about 30 type 2 diabetes GWAS signals by then, which are represented here. But we also had this parallel effort led by Anish Barroso and others on MAGIC, which was looking at glucose and insulin levels in non-diabetic individuals and giving us readouts, therefore, of beta cell function and insulin resistance. So we could start to layer that on top. So this is showing those diabetes hits on a scatter plot where the axes are beta cell function and insulin sensitivity. And this really nicely showed that not all diabetes signals were alike. They weren't all leading to diabetes through the same mechanisms, that some of them were working predominantly through obesity or insulin resistance in the top left, and others presumably through direct effects to compromise beta cell function. And further efforts to show that you could, by overlaying open chromatin maps and expression data, you could further sort of differentiate and corroborate some of those findings, and that ended up in that recent paper in Nature from 2024, being able to take those 1,000-odd loci and cluster them into 8 different groups, which have differences in the pleiotropy across other phenotypes, differences in the cell type enrichment, and for the first time, really some differences in the clinical outcomes that come from people who are differently loaded for these different factors. And then finally, the final lesson is probably the most important, which is that people matter and trust and respect matters. And I think one of the reasons why the grouping with Mike and David and myself and many of you was so sustainable was because of Mike's position as the sort of stable core of all of this, the dependable core of all this, bringing us all together, making sure that we behaved in as far as possible in the right way, while maintaining momentum. I think one of the challenges we had great groups, great folks in Mike's group, and great folks in David's group, and great groups folks in our group. And these are all postdocs competing for papers, and then we were doing these big science papers where we had hundreds, sometimes even thousands of authors on that, and navigating that wasn't always easy, particularly when we'd arranged to send 3 papers in to a journal, and then the journal said, yes, we'll publish it, but only if you bring them together into one paper, and you can imagine what traumas that caused. So it took somebody of Mike's trustworthiness and diplomatic skills and EQ to help us navigate that. So incredibly grateful to Mike for charting the course there. So let me just finish off. Way too many acknowledgements to mention, but I did want to just call out some of the folks from New Michigan Stable that I've worked with and over the years, and it's been a fantastic interaction, but also to call out the many other folks who've contributed to this work, some of whom are in the audience. It was just great to have this opportunity to just review the last 20 years and think about some of these things. So thanks, Mike, for the opportunity, and thanks for everything that you've done.